Welcome to the Explainer. Today we are unlocking the vibrant, totally hidden world of plant pigments. We're going to dive deep into a complete thin layer chromatography, or TLC experiment, specifically following the fantastic procedures presented by Dr. Kem. You know, we look at green leaves every single day, right? But hiding just beneath that uniform green surface is an absolute symphony of complex chemical colors. Our goal right now is to walk through this complete TLC lab experiment together, from the foundational chemistry all the way to the final calculations. Okay, let's dive into this. Here is our roadmap. We'll start with the theory of TLC, move into materials and sample prep, go through the actual experimental procedure, break down the observations and calculations, and finally, look at the results and applications. First up, Section 1, Theory of TLC, Unlocking the Chemistry of Color. Before we get our hands dirty on the benchtop, we need to understand the why. So, what exactly is TLC? Well, simply put, thin layer chromatography is a really convenient analytical technique used to separate out the components of a mixture. It basically works by exploiting a chemical tug-of-war. We've got a solid stationary phase, usually a piece of plastic or aluminum coated with a super thin layer of silica gel, and a liquid mobile phase, which is an organic solvent. Complex organic molecules, like our plant pigments, get separated based on their varying affinities for these two competing phases. You can kind of think of it like a giant chemical obstacle course. You apply your mixture as a tiny spot at an origin line at the bottom of the plate. As the mobile phase solvent moves up the plate through capillary action, it pulls our little pigment suspects right along with it. And based on their unique chemical properties, some of those pigments get dragged super fast to the top, while others kind of drag their feet and stay near the bottom. That's what reveals their true colors in distinct bands. Now, the really crucial point here is understanding why they move at different speeds. It all comes down to polarity. The silica gel stationary phase is very polar. That means hydrophilic or water-loving compounds have a huge affinity for it. They stick incredibly tightly to the silica, so they move very slowly. On the flip side, nonpolar hydrophobic compounds have a high affinity for the organic solvent mobile phase, so they practically sprint ahead with the solvent front. Moving right along to Section 2, Materials and Sample Prep. Let's equip that benchtop. To properly extract these pigments, we absolutely need the right tools. We're going to start with 250 milligrams of fresh leaf tissue. Standard spinach works great, or something like copper leaf. We also need our chemical keys to actually unlock those pigments. 90% acetone to solubilize everything, along with hexane and water for the separation phase. We'll be using a mortar and plastic pestle for grinding, a microcentrifuge to spin down the debris, standard TLC silica gel plates, and glass capillary tubes for a really precise application. The extraction procedure itself is pretty much a precise four-step dance. First, you grind up that 250 milligrams of leaf tissue with 400 microliters of 90% acetone to break open those plant cells. Then, add another 600 microliters of acetone and give it a really good mix on a vortex mixer. Next, pop those tubes into a centrifuge at 10,000 RPM for 3 minutes to separate the solid plant junk from the liquid we want. Finally, transfer that liquid supernatant to a fresh tube and mix it with 150 microliters of hexane and 200 microliters of water. This is the magic step. It forces the lipid-soluble plant pigments to literally partition right out into the hexane phase. Okay, so with our concentrated pigment extract ready to go, we prep the TLC plate. Precision is absolutely everything here. You have to draw a faint line exactly 1.5 centimeters from the bottom edge of the plate, and you must do this with a pencil. If you use a pen, the ink will run, and if you press too hard, you'll scratch that delicate silica surface, which completely ruins the upward capillary flow of your solvent. Section 3, the experimental procedure. We are ready to run the test. Now for the main event. Using a glass capillary, you very carefully apply tiny aliquots of your green extract directly onto that origin line until you've spotted about 10 microliters total. You gotta let the hexane evaporate completely between each application so the spot stays super small and concentrated. Once the plate is bone dry, you place it upright into a pre-saturated chromatography chamber. The solvent will immediately start climbing up the plate. The second the solvent front almost reaches the top, pull the plate out and immediately mark that final solvent line with your pencil. I do need to mention that the specific chemistry and the safety profile of our mobile phase is critical here. We are using a 3-to-1 ratio of petroleum ether, acetone, and chloroform. Because these specific organic solvents are highly volatile, they smell pretty awful, and they carry health risks, like causing dizziness. This entire developing step must always, without exception, be performed safely inside a functioning laboratory fume hood.
All right, section 4, observations and calculations. Let's make some sense of all this data.
So the solvent evaporates, and suddenly you're left with this gorgeous rainbow of distinct bands right there on your plate. But beautiful colors aren't really data, right? So what does this all mean? How do we turn these floating, subjective bands of color into objective, hard data that a lab in New York can perfectly compare to a lab in Tokyo? The answer is actually this incredibly simple, powerful formula called the retention factor, or RF value. You calculate it by taking the distance traveled by a specific pigment compound and dividing it by the total distance traveled by the solvent front. Now, because the solvent always travels further than the actual pigments, your RF value is always going to be a decimal somewhere between 0 and 1. To actually get these numbers in the real world, you just grab a standard ruler. You measure from that 1.5 centimeter origin line we drew earlier straight up to the exact geometric center of a pigment spot. That number is your numerator. Then you measure from the origin line all the way up to the uppermost line where the solvent stopped. That's your denominator. Just divide the two and boom, you have your standardized RF value.
And when you do the math, it is so cool how perfectly the visual sensory data aligns with the chemistry. Just look at beta-carotene. It is incredibly hydrophobic, so it practically sprints up the plate, giving us this distinct orange band with an RF value of 0.97. But contrast that with anthocyanin. It's water-soluble, it's polar, so it stays stuck right down near the bottom of the silica plate, giving a red band with an RF of just 0.10.
And this brilliantly illustrates exactly why these molecules behave the way they do. Look at the complex chemical structure of chlorophyll A here, especially that central magnesium ion. Its specific arrangement of bonds and its overall polarity dictate exactly how it's going to interact with the silica gel and the solvent. That's what gives it its dark blue-green visual signature and a very specific RF value of 0.79. It is quite literally molecular structure determining macroscopic behavior. Which brings us to our final section, Section 5, Results and Applications. Let's explore some advanced techniques. Let's see how this all builds into what chemists call reversed phase chromatography. So everything we've talked about up until now, that was normal phase, a hydrophilic polar stationary plate combined with a nonpolar mobile solvent. But in reversed phase chromatography, we flip the entire script. We use a hydrophobic modified stationary plate, like RP18, and we pair it with a highly polar solvent mixture, usually containing things like ethanol and acetronitrile. The visual proof of this concept is absolutely stunning. Just look at this side-by-side comparison. On the left is the normal phase, with our nonpolar carotenes racing to the top. But on the right, in the reversed phase plate, the literal sequence of the bands completely reverses. Now the polar yellow xanthophylls are moving to the top, while the nonpolar carotenes are the ones held back at the bottom by the hydrophobic plate. It's the exact same leaf extract, but it's revealing its secrets in a completely different way, based entirely on the chemical environment we designed. You know what is truly amazing about this specific TLC method from Chem? Its incredible efficiency. Whether you're running normal or reversed phase, clear, vivid separation of these vital biological compounds is often achieved in less than 8 minutes once that plate goes into the chamber. It is just such a cost-effective, time-efficient way to peek into the chemical makeup of life. So as we wrap up this explainer, I really want to leave you with something to ponder. We have just used some basic laboratory glass and simple organic solvents to literally pull apart the hidden molecules that capture the sun's energy. If a simple TLC plate can reveal the vivid, unseen architecture of photosynthesis hidden inside a basic spinach leaf, what other vast, unseen chemical pathways are operating right now, quietly driving all life on Earth, just waiting for us to discover them? Keep wondering and keep exploring.